Welcome to the virtual World Stem Cell Summit 2022. I'm Dr. Jane Andrews, the founder and CEO of CellBridge Strategies, which is an international growth consulting firm that focuses on the life sciences and regenerative medicine. My team helps companies match their strengths with market opportunities by bridging the gap between science and business. This is often very important because it involves helping companies with targeting their key assets, creating strategies, executing on tactics, building financial and market models, and in many cases, identifying potential partners, funding opportunities, or M&A capabilities and opportunities. We are honored to be selected as the premier consulting firm for Bernie Siegel's virtual World Stem Cell Summit 2022. Today, we'll be presenting a talk and panel discussion entitled Living in Your World from Bootstraps to Market, Navigating the Multidimensional Roadmaps. But first, before I begin with the talk, I'd like to introduce our panelists today that are all part of CellBridge Strategies. Preveen Ramamoorthy. Preveen? Dr. Preveen Ramamoorthy is an advisor and principal consultant for CellBridge Strategies. He is an R&D scientist and executive with 2 decades of product development and leadership experience in cell and gene therapy, regenerative medicine, and development of biologics. His area of expertise crosses many disciplines, including precision medicine, global health, and diagnostics. Mike Fiske. Mike Fiske is a principal consultant for CellBridge Strategies and the VP of a biomanufacturing Cdmo in San Antonio, Texas. Mike is a highly motivated scientist and executive with significant experience in early phase development of protein and cell therapy products. He has a proven track record for candidate discovery, characterization, and process development through clinical scale Cgmp production and testing. His technical background and expertise span a wide range of biotechnical products, including the advanced therapies and vaccines, industrial enzymes, metabolic, pathway intermediates, diagnostics, and therapeutic proteins. Keith Parent. Keith Parent is a partner of CellBridge Strategies as well as an advisor and principal consultant. He also is the founder and CEO of his own company called the Court Square Group. It's a cloud-based IT company that focuses on FDA-compliant documentation. The company also provides IT integration platforms with a single user-friendly interface. Keith is also the co-founder and partner for RegDocs 365, which is a powerful suite of tools that enables life science startups and CROs to use the same tools as Keith's Fortune 100 clients at a very affordable rate. Neil Kellan. Neil Kellan is CellBridge Strategies CFO and principal consultant for corporate strategies. His 25 to 30 years of experience has given him a unique ability to leverage diverse perspectives to create innovative strategic plans for cell therapy, biotech, and biomedical companies. In addition to corporate strategy, Neil has also developed competitive and price positioning strategies across the globe for one of the top 5 pharmaceutical companies in the world. In this case, he built a quantitative model encompassing global demand, capacity, production, distribution, and packaging while working in numerous market segments. He then made recommendations to the client based on his findings on preferred niches and competitive tactics. Neil has also led and been deeply involved in multiple M&A initiatives over his career. I am very thankful to have a team such as this supporting us at CellBridge Strategies. The diversity of the expertise allows us to help our clients do what I think we do best, which is bridge the science to the business. And that's what it really takes to be successful in this space. So the title of the talk today is Living in Your World from Bootstraps to Market, Navigating the Multidimensional Roadmaps. First of all, I'd like to just go over who we are. We're CellBridge Strategies, and we understand your science and its implications for business vision, strategies, and tactics. We're a global growth consulting firm that focuses on the life sciences and regenerative medicine. We help biotech and life science companies align their operations and strategies, accelerate product development, reduce costs, help them with their return on investment, and really move forward in the track that allows them to commercialize their product. We customize each product that we offer for the needs of our clients. And who are our clients? They range from life science, any area of life science, pharmaceutical companies, to CDMOs and CMOs. So we're going to start to talk about the multidimensional roadmaps and convergence of technology that is needed for regenerative medicine and the advanced therapies. And this can get rather complicated, but what I'd like to focus on right now before we begin this section is that the convergence of numerous technologies and disciplines are required for both let's 1, 2, 3, 4. Regenerative medicine and the advanced therapies is an ecosystem that is very complex. It is a convergence of numerous technologies and disciplines, requiring both broad and deep expertise. CellBridge Strategies can maneuver through the ecosystem to identify the winning combination of roadmaps. We first take into consideration the science because that is really the most important thing. Without the science, you have nothing. But without the business structure, it's impossible to make the science into a product. We marry the science and the business to help our clients with vision, strategy, and tactics. So the business thesis is really something that CEOs must consider. They must align the science and the business strategy. The biggest challenge for regenerative medicine business leaders is building the bridge between exceptional science and exceptional business. So what is all of this multidimensional convergence? Well, regenerative medicine really comes together because of the convergence of scientists, engineers, technologists, intellectual property, information technology, all working together to address the new phase of medicine, which is regenerative medicine cell therapy, or the advanced therapies, however you want to term it. The convergence of all of these different areas requires an entrepreneur to navigate these new technologies and the highly complex systems that exist. CellBridge Strategies understands the convergence of these multi-dimensional technologies to help our clients become successful. So why is it important? Because number one, investment is pouring into regenerative medicine and the advanced therapies to the tune of billions of dollars. Well, why is that happening? That's happening because we are building the next pillar of medicine, that being regenerative medicine and the advanced therapies, because these therapies can truly cure patients. Now let's start talking about these roadmaps. This first slide is rather complicated because it really is all of the roadmaps put together. And what I'd like to emphasize here is that we have to think about what happens from collection of the cells from the patient or donor, how those cells are processed or changed and grown, how they're frozen, how they're thawed, and how they're transported back to the patient. So it's really an arm-to-arm approach. When the cells are collected from the patient or the donor, they're usually collected through a process called apheresis. which involves standard operating procedures. Well, many apheresis centers, for example, are still doing this with pencil and paper rather than doing them through IT and the cloud. This really introduces a lot of variability, even at the very beginning of the whole manufacturing process. So getting cloud-based systems in place at this time is very important, and it also is needed to ensure HIPAA compliance. When we go further, we start to look at whether or not the cells are modified genetically or not. You must have the right source of vector. You must have that supply chain in place and locked down. Once the cells are modified, or even if they're not, the cells must further be processed. It's important to test these cells in process to find out if they're still healthy or potent. And if they're potent, are they producing enough of the substance that you want them to produce to be an effective product and cure patients? Then they start the expansion process. And this is usually done in a bioreactor, but more recently, companies are starting to expand in patients, which is a very interesting new approach. But before you were to put these cells back in a patient, you'd need to test them, have QC and QA processes in place to support the process, so you know the cells that you're putting in the patient are of the highest quality process for treating the disease. Once expansion is complete, whether in the lab, if it's in the lab, those cells must be stored for subsequent treatment within a short period of time, or stored for treatment of that patient later on. Oftentimes they're cryopreserved when they're stored. So looking at those cells before you put them in the freezer or afterwards when they're thawed is very important. Then looking at how those cells are transported back to the patient, where the patient is going to be treated, whether that's the hospital or the medical center. Numerous processes with IT and tracking of those cells, the temperature at which they're exposed to must be in place for logistics to be successful. And not only that, really transposed on top of all of this is the payer access. Who's going to pay for it? How much is covered by insurance? And the heart of all of this is government regulation. So before any of this is possible, we have to make sure that we're compliant with FDA regulations as we develop the product. Then we have all the support industries that support this as well. And we'll get into that as we go further. Source materials, I spoke about that briefly. Source materials involve assay kits, apheresis, vectors and plasmids, chemicals that are Cgmp compliant. Oftentimes vendors will say their product is CGMP compliant, but it's really not certified as CGMP grade. You have to be very careful about that. Culture media also needs to be CGMP compliant. Disposables must be. Equipment must be validated. Cryomaterials must also be CGMP compliant. It's important to know the source of your materials, how really CGMP compliant they are, the quality of those materials, and lock down long-term contracts, because right now there's a huge shortage of source materials for the industry. This all must be considered to bridge that science to business. So this is something that the executive team and operations must be concerned about as they move forward, even before they create a product. Information technology and artificial intelligence ties it all together. As you're a team moving forward in product development, you must think about the IT that you're going to use, whether it's cloud-based, how it's going to capture patient information and keep it HIPAA compliant. The source of the source materials must also be documented through information technology. Your quality management systems are really important. The more manual they are, the more expensive your COGS are. So having that as automated as possible is also important. Process engineering. Sometimes a product is not ready to become a real product to treat patients. It must be process engineered to scale up. Manufacturing must be considered, logistics, payer access, all of this must be integrated with information technology. And Keith will talk more about that and artificial intelligence and how that will support the industry. Product testing. I briefly spoke about that in the beginning as well. The cells must be tested at the time they're recovered from the patient or the donor for viability. Are they alive? How many of them are there? Must be evaluated in the very beginning. And then as you go through the process, in-process testing must be also taking place for identity, purity, quality, and as well as safety. Are there any contaminants in with the products that would be dangerous to the patient? Inline testing must take place throughout product development, and then potency must be definitely approached and identified at product release before it goes into the patient. Biomanufacturing is the biggest gap in the industry today. There is a shortage of biomanufacturing facilities that are Cgmp compliant, and therefore they are stemming up all over the world. Being able to qualify a facility for your use is something that's incredibly important, and Mike Fiske will speak about that in more depth. How do you know which Cdmo to pick? How do you know which one is going to help you through the entire process? Does it have the capacity to take you through clinical phase 1, process engineering, all the way through commercialization? Or are you going to have to switch in the middle? There are many things that must be considered as you work your way through the selection of a biomanufacturing facility. And if you're doing it in-house, you need to consider all the things that a Cdmo would consider. Personnel and corporate structure. This is kind of where I feel the rubber hits the road, because you have to have scientists as well as executives interfacing with each other, where we bridge science to business. One needs to consider, who is the executive team? Are they qualified? Do they have enough business experience to run the company? Who are the engineers that are helping engineer this technology into a viable product? Who are the biologists and physiologists? What physicians and nurses will be involved? What technologies, IT technologies will be used, and what quality management personnel are going to be needed? Finally, when you have this product, how are you going to take it to market? What marketing strategies are you going to have? And what regulatory, could you have a regulatory person on, or are you going to need to have a regulatory consultant? And then who are you selling to? Are you selling to the hospital, the medical facility, or direct to patient? And is this covered by insurance? All of these things need to be considered, as well as whether you're going to take this product all the way through to commercialization, or you're going to take it to clinical trial phase 1 and then sell it off to someone else who will carry it down to commercialization. All of these aspects need to be thought about by the executives and the operational staff moving forward. That's bridging the science to the business. Finally, we have roadmap number 6, which is indications and markets. So as you can see, each one of these indications have markets that are in the billions of dollars. So how do you select which indication you're going to go for clinical trials? So if you have a product that really has use for metabolic diseases, you may want to focus on diabetes, because that is a huge market. But there are a large number of competitors in that space, and it may take you a very long time to get FDA approval if you go with diabetes as your indication. Therefore, you may want to start with a smaller indication that may be an orphan disease, because you may be able to fast track your product through FDA approval that way, and then subsequently pivot to a larger indication. That's one example of some of the things that you need to consider as you work your way through FDA compliance and approval. CellBridge strategies can help you as you work your way through these strategies. Product and cell type. As most of you know, there's numerous cell types and products out there. Cells can vary all the way from immunological cells, such as T cells and NK cells, to numerous stem cell types. Hematopoietic stem cells from bone marrow or perinatal tissues. Perinatal tissues can be also very high in MSCs of high quality that are immunoprotected. You have adult stem cells from adipose tissue. There are other sources of MSCs as well. And then you have induced pluripotent stem cells that have a lot of potential in the market as well. And then pluripotent stem cells, that of embryonic origin. So thinking about all of these cell types and how they might be most useful for your potential technology and reaching the market is very important. That's very early stage. CellBridge strategies can help you select the right strategy and cell types for moving your product forward here as well. Patient access. This is something that is a moving target for the industry, especially in cell and gene and in cell therapies as well. But number one, we have to think first of all about how the patient is going to be treated. How are you going to get access to the patient? Is the treatment going to be used as a first level treatment, first, second, or third level? You need to consider that as you develop your product. Then you need to think about the pharmacoeconomic analysis that usually needs to be thought about. Once you have some proof of efficacy, you'll be required to do a pharmacoeconomic analysis. Very early on, you need to think about the therapy pricing. This will allow you to understand what your return on investment will be and who your competitors are, what they're selling similar products for, and whether or not you'll be able to make it in the market at the volume that you can create. Access to electronic records and blockchain often is very important. You need to keep those records secure. How are you going to do that? Insurance provider hubs. Oftentimes patient access is integrated with the providers. You have to think about patient out-of-pocket expenses, payer approval, treatment orders. Who is going to order it? How is the doctor going to call and order your treatment? Is this going to go through the hospital pharmacy? How is that going to occur? All of that needs to be considered. And then how knowledgeable is the patient about the product that you're offering? And then who is going to administer it to the patient? All of this is part of patient access, something that you need to think about early on, or executives in companies need to think about relatively early on. So the regulatory pathway can be very complex, and it often depends on the product, the technology, and the indication that you choose. This will then dictate the CMC requirements, the IND status, and whether you're able to fast track through something like RMAT, breakthrough therapy, or orphan drug access. Accelerated approval can be facilitated a number of different ways, and this is something that Mike and Preveen are much more familiar with, and we can assist in mapping this process out for the companies involved. So in summary, there are many winning combinations through each one of these roadmaps, and the roadmaps are in the squares around the wheel. But there's more than one way to succeed in this industry, but you must select the appropriate pathway through the roadmaps. And that is what is depicted here. And we have the capabilities to help you along the way. So in summary, opportunities abound. Regenerative medicine has shown that it can cure patients. Manufacturing capacity is in great demand. Huge opportunity is out there to reduce costs and increase return on investment through automation. Source material supply chain can become well developed. The labor pool has not yet caught up with demand, but as we focus more and more on training of individuals, we will have a labor pool to support the industry. Logistics strategies are currently being developed, and infrastructure is immature, but then again, it is developing as we speak. Insurance approval mechanisms are in flux. Patients are waiting to be cured, which means there is an unfulfilled market demand. There's a substantial clinical trial pipeline, and markets are very fluid and currently undefined. We firmly believe that marriage between the technology companies and the support industries will be necessary to advance this industry, regenerative medicine and the advanced therapies. IT now and artificial intelligence for the future will be necessary. Manufacturing capabilities and capacity must be expanded. New assays are being created as we speak. Methods for new cryopreservation protocols are being created. Logistic technology is being honed, and product delivery to patients using software, medical devices, and IP for combination products are also in development that will support the technologies that will cure disease. So what are the implications of the roadmaps? For entrepreneurs, the value of companies to be acquired depends on the ability to handle the convergence across dimensions, business and technology. For funding agencies, it's important to invest in companies that have an inherent potential to work together. Product companies, which are technology and science, service companies, which are IT, logistics, product delivery companies as well. If you can knit together individual companies to create masteries across dimensions, you will create a whole that is greater than the sum of the parts. To be successful in this very complicated, divergent, multidimensional industry, you have to have the ability to make decisions rapidly at the level of the executive team. You must be flexible and be able to pivot when needed. And you also have to keep your fingers on the heartbeat of the industry, your competitors, and your customers. In fact, we, CellBridge Strategies, can be your owl to help you as you navigate your way through these systems. Our team has over 125 years of experience working in many facets of this industry, and we're here to help you build your business from your science to cultivate cross-silo leadership and collaborate to advance your business goals. And here's the team that is present to talk to you today in the panel discussion that will be next. Neil Kellen, Keith Parent, Mike Fiske, Preveen Ramamoorthy, and myself. Thank you. So I'd like to start the panel discussion for CellBridge Strategies, and we'll start with Keith Parent. Keith is a partner, advisor, and principal consultant for CellBridge Strategies, and he's also the founder and CEO of Court Square Group, a cloud-based IT company that focuses on FDA-compliant documentation. He's very familiar with the IT needs of the industry, and I'm very excited about learning about his perspective on what our clients are often asking us in this industry and how we can serve them. Keith, welcome. And thank you,
Jane. Thank you, Jane. It's a pleasure to be here. I'm excited to be part of this panel, and your presentation was fantastic. Some of the questions that we get asked are things like, how does IT within the cell and gene therapy world differ from large pharma, or does it? And when I think about where the systems are today in the cell and gene therapy world versus where large pharma was, maybe 10, 20 years ago, those systems have now come into and are very mature. And some of those systems are being used for some of the cell and gene therapy world, but they're not really applicable for that. So they've kind of have to morph themselves a little bit for the cell and gene therapy world, whereas there's a lot of startup companies with some software in the cell and gene therapy world that are actually making a difference and you're working end to end. So I think there's some immature systems out there that are getting better. And I think that we just have to look at how they work together. There's lots of independent systems that we try to integrate together when we're pulling those out. When you think about all of what you talked about and all the different systems around each of the charts that you had out there, there's always IT involved in those systems. So part of the goal is to figure out where are the integration points between all of those. There are some companies that are trying to do all the way from the beginning all through the very end, but that's going to be very expensive. And part of that's going to be the cost and the capital outlay that these companies would need to deal with. So you have to understand where that comes in. A question that we get asked are, at what point do we start dealing with data systems in our process? And realistically, you know, I'm a big pusher on our companies of looking at process first and understanding how the process is out there. But I believe that you can't start collecting data soon enough. I think there's lots of data that you have. Some of the first data that you're going to collect are even your standard operating procedures, and putting those down on paper so that you can get those, and we'll automate that actually. But let's get the process down, understand how it works, and put it out there. When we talk about automation, this goes hand in hand with the last question about grabbing data. Typically, we're going to want to automate the more we get data and the more data that is going to be generated as part of that process. The reality is we're going to want to have a lot of the data in the systems that are out there. 1, 2, 3. When I think about automation, part of what we're looking at is the amount of data that we're going to be collecting and gathering a lot of that data through automation, because we don't want to be gathering that manually throughout the process. However, one of the big problems that we certainly have is if you don't really understand your process and we try to automate a bad process, that's just not going to serve yourself or serve anybody any well. The goal really be to look at the process, understand how to get that down first in the lab and make it work right, and then automate that process so that we can understand how to get it to the next level. And then the data that's generated that is actually very usable data. Our goal from that perspective is to understand each of the different segments of the process. And do I need to have one system that encompasses everything? Or do I have individual systems that take care of each of the different processes along the way? With that in mind, another area that we think about in this particular industry is certainly, if I'm a patient and you're taking my cells out and you're going to do something with my cells and put them back into me, and it's a needle process, I want to make sure that I get those cells back in my body after you've done something with them. Blockchain is a huge enabler of us looking at making sure that your data gets back to you or your cells get back to you. So using blockchain to track and trace all the way through that process is a huge technological advantage. You know, we hear about blockchain a lot in the financial world, but the reality is those same techniques used in the financial world to track your transactions for your money can actually be used to track your own cells throughout the process. So blockchain is a very big piece of how we're going to pull some of that together and drive a lot of the traceability aspects of the different systems, because your data is probably going to run through multiple systems all along the way. And we're going to want to be able to understand how that data is fed together throughout that process. Can I use cloud-based systems to interface with both my lab and my manufacturing environment? You know, most companies now are looking at, can I put together a cloud-first strategy? And how does that work? When I talk about a cloud-first strategy, what does that mean? That means, how do I not have to build out a whole new data center and to put everything in my plant? Can I use an internet connection to come out to my environment and be able to gather data along the way? Part of that is a function of the amount of data that's going to be collected along the way, and then how much data is going to be processed either internally or externally. So part of what we do is we look at the systems themselves, understand where the data is going to be collected, or if it's going to be analyzed. You may actually look at a lot of companies that have software that can use it to analyze that data and pull that data out from the different systems that they have and use that along the way. So part of what we do is we talk to companies around, have they already bought or purchased other systems that are actually on-premise based systems, and can they integrate with a cloud system? If they can, then let's go that route. Otherwise, if it's a fresh new company that's just starting out, we try to look at, can we do more things that are cloud-based? Or if my plant is in an area that's that doesn't have great internet connectivity, is there a problem with something that could potentially be totally cloud-based and then has spotty internet access? So the goal would be to look at where you are, what you're doing, and how you're trying to pull some of those systems together. That would be a big area of concern, particularly for a company that may be in a foreign country that's talking to something, maybe an analytical services back here in the US, and you have to deal with the time differences. Another big aspect that we've talked about, and you alluded to, Jane, in the presentation, was the concept of AI and how does AI affect cell and gene therapy. One of the areas within AI is the concept of using machine learning to figure out, can our processes get better along the way. With the volumes of data that are out there, it's actually a prime area that machine learning can look at your processes, look at the data collected as part of that, and either look at safety parameters or different aspects of a process and be able to help us hone down that process to eliminate some of the issues that you had actually alluded to, whether it's during the manufacturing process, during the analytical process, or beyond. That's just a smattering of a few of the things that we're going to deal with from an AI perspective or from an IT perspective. I'll contribute later on, but I'll let the other people go now and see if there's other questions that we have.
One of the things that I'd like to point out is that Keith and I work very well together, and that we often work together to map the science and the technology to the IT. Because there's such diversity out there in what those requirements are, we can facilitate each other and our capabilities by working together in that respect. Mike, did you have something you might want to add to that?
Oh, that was extremely well said. And I'll wait till my session to talk about some additional questions.
Okay, all right. Preveen, do you have any thoughts on IT and?
Yeah, compared to traditional biologics, cell therapies are very, very complex because you're dealing with the cell, you're not dealing with the antibody, a monoclonal antibody. So the amount of data and information that flows into the system, in terms of when you talk about autologous therapies, is a huge amount of analytical data, clinical data that flows in. And machine learning becomes a really important way, and blockchain and machine learning become really important tools in which we can, help, clients organize themselves and be able to derive value out of their own data.
Thank you. So one of the other things I often think about with regards to this is that, you're collecting patient data, you're collecting assay data, results from flow cytometry, from HPLC, or whatever testing process you're using to test the cells along the way. It's complicated equipment that have their own analytics and their own data processing systems often inside the machine. And how do you grab that information and put it into another system that's readily accessible through a user dashboard? And I think that Keith partners with a number of different companies to facilitate that process. I mean, we're partners to facilitate the mapping of the technology, but not only that, the RegDocs 365 helps as a dashboard, it's my understanding, that helps to bring sort of a key and lock a combination of different systems in so that it can be looked at in a user-friendly way. Is that correct, Keith?
Yeah, it's actually really a platform for gathering all the content from the different processes along the way and be able to display that back to an executive team to make informed decisions about what's happening along the way, whether it's early in the process, later in the process, during the supply chain aspects. There's lots of different areas that are of concern, particularly in this industry. And our goal is to put something out there that stays regulatory compliant, but still gives you the ability to see the data that's most pertinent to whichever aspect, whether you're regulatory, operations, financial, all those different pieces.
Yeah, that's a very important thing. And I don't find a lot of other companies that do that in a way that makes it affordable for startups. Many of the systems that would be competitors they're hundreds of thousands of dollars for systems. And I like the idea of making some of these processes available to startups with a starting cost, and then they can build from there. Because with other companies, they're buying things they don't even need yet. So with Court Square, they're buying what they need: 1 chair, 2 chairs, whatever it is.
Well, I'm a firm believer that you may have already invested in certain applications. Why don't we use the best of what you've already got and see if we can use that in conjunction with other applications that may be out there, or use a best of breed approach toward what you're trying to do, depending on the process that you're trying to incorporate.
Thank you. Thank you. So I
Yeah,
think we'll go on,
Jane,
Mike.
yeah, let me add one thing, because as Keith was just talking, it prompted me to actually generate a good thought or a question about this. You mentioned the compliance aspects of it from a regulatory perspective. I think that's, being a manufacturing person, that's very important to me, to make sure that these types of systems are validatable, and that they do comply with global regulatory requirements wherever you may be marketing products. So yeah, that's a big point.
Thank you. Thank you. So without further ado, we'll move on to Preveen. Preveen Ramamoorthy is one of our advisors and a principal consultant, and he has just such an expansive background. I find his information, his knowledge to be just invaluable, as him and I often brainstorm almost on a weekly basis about some of the projects that we do. So Preveen, if you'd like to take it away and talk about some of the questions that clients ask you.
Thank you, Jane. Thank you for that kind introduction and that wonderful presentation that you had earlier. So, this whole area, as Jane alluded to earlier, this whole area of cell therapy and regenerative medicine, and also Keith also made a remark that, it is where the biologic space, the monoclonal antibody therapeutic space was, 1 or 2 decades ago. So there is just so much that this field has progressed, but it needs to progress even more, because the innovation is very high, but how we translate those innovations into actual therapies, because of the sheer complexities of these cell therapies, is the challenge ahead of us. So in that respect, there are certain 3 challenges, I would say, that I wanted to highlight in this panel discussion that I've heard, clients talk to me about. One is, what are the challenges in characterizing, cell therapies? This is one of those central challenges that, regenerative medicine and cell therapy companies face. And then companies that are pursuing autologous cell therapies, face their own unique challenges. So that's the second one. And the third one would be, what sort of applications and indications can we take pluripotent stem cells into, and so these are the 3 challenges that I wanted to sort of elucidate further, in this panel discussion. So let's start with the first challenge that I articulated earlier: challenges in characterizing, cell therapies. And this sort of ties into roadmap 3 that Jane presented. Unlike monoclonal antibodies or protein therapeutics, which are essentially single molecules, or even viral vectors or viral vaccines, cell therapies are exceptionally complex due to the sheer diversity, of their genomic, proteomic, metabolomic composition and activities, and their physiological activities in vivo. Because you've got a whole cell in a, doing its thing, as opposed to a single molecule or a virus. So there you can actually appreciate, why is it that we're saying that these are so complex, and characterizing them is indeed a challenge. So how do we characterize these? We use analytical assays to characterize these cell therapies. And you can, as Jane alluded to earlier in roadmap 3, you can sort of look at the character of a cell therapy, with identity assays. Those are assays that tell you, okay, this is exactly the identity of the product that you're going to infuse into a patient. You can be looking at gene expression markers. You can be looking at cell surface markers in order to distinguish your product. The next one would be purity. Along as you manufacture, you will get some contaminating cell types potentially, in the, amidst all the majority of the cell therapy product. You want to be able to assess, what is the purity of it and what are the other contaminating cell types, the impurities in it. Thirdly is potency. And this is really the biggest challenge of all in these cell therapies. And what is potency? Potency is a measure of biological activity that can predict clinical efficacy. So this is a really challenging one, as I said earlier. The central challenge is that you're trying to use an in vitro assay to predict the in vivo efficacy of a highly complex biologic such as cell therapy. And so one assay, a potency assay, may not cut it. So you may need a matrix of assays because it's so hard to select one assay that is going to predict how this in vitro, the cell therapy that you're testing in vitro is going to behave in vivo. So this is one of the biggest challenges I would highlight relative to the others. Not that the others are any less challenging, but this is one of the biggest ones. Safety. These assays are fairly straightforward in the sense that they are commonly used to test for in the biologic space or in the vaccine space. Like you look for adventitious agents, you look for mycoplasma, you look for bacterial contamination in the form of sterility testing, you look at endotoxins. And safety can also, in the case of pluripotent stem cells, can go into tumorigenicity assays and such. And then there are, of course, other assays such as viability and cell counts and appearance. So that sort of summarizes sort of the first challenge I wanted to sort of highlight, the characterizing cell therapies part. Now let's get into what Jane talked about in roadmap 4, is the manufacturing challenges. And this is what we have particularly with autologous cell therapies. The manufacturing challenges are very pronounced, and Mike Fiske, I'm sure, will have a few comments or details going into that aspect. But I wanted to sort of set the stage, for what are the challenges of using autologous therapies. We all know how amazing CAR T cell therapy has been. And we have already seen the first patient, 10 years cancer-free, Elizabeth Whitehead. And she runs this Whitehead Foundation, which is amazing. I've had a chance to meet with her when I was at Terumo. A very, very inspiring story. So practically, one can say that, with 10 years of remission and absolutely no detectable cancer, one can say that we are close to a cure with CAR T cell therapies. But this is an autologous cell therapy which has shown tremendous clinical benefit. Therefore, one can't say it's difficult to manufacture, therefore we shall not go that route. So I don't see autologous therapies going away anytime soon because their clinical impact has been phenomenal. So what do we mean by autologous cell therapies? Simply put, autologous cell therapies are therapies in which the cellular or tissue material originates from the patient that needs to be treated. If you contrast it with allogenic therapies, the cellular and tissue material does not originate in the patient that needs to be treated. So the fact that the cellular material or the tissue material originates from the patient that needs to be treated creates a lot of challenges. And as I already alluded to earlier, manufacturability is an issue because you have to control the product consistency, and that can be very challenging because every patient is different. Every patient's cancer situation might be different. Therefore, the cellular tissue material that originates from one patient is not exactly the same as the cellular material or tissue material originating from another patient. So manufacturability becomes a challenge. The input material to manufacturing creates those manufacturability challenges. So the other thing is the higher burden of analytical testing and lot release assays. Now, if you imagine an allogenic therapy, which you manufacture a batch of a cell therapy that can go to 10 to hundreds of patients, one batch requires a battery of analytical testing and lot release testing. Whereas with autologous, one patient's material is one batch, so the very same extensive battery of release testing that is done to a biologic product batch or a batch of allogenic cell therapy, essentially needs to be done for every patient. And then you also need a very third point is you need a high level of expertise to manufacture. Why do I say that? Because we don't have exactly the most standardized end-to-end systems to manufacture autologous therapies, although they are coming into the picture. There is a tremendous amount of human intervention, high complexity intervention and expertise that is needed to manufacture these autologous products relative to when you think about manufacturing a batch of viral vaccines or viral vectors and all of that. There's a lot of babysitting that goes on, from deriving the material from the vein of a patient in a hospital and all the logistics that comes along with it, and taking it into the manufacturing facility. And typically not just one instrument is used, multiple modular instruments are used from culturing and expanding and transducing these cells with viral vectors and concentrating them and fill finishing them and whatnot. So there's just a lot of instrumentation and expertise that is needed. So the fourth point is the complexity of logistics. You can imagine you've got a cancer patient very sick in a hospital with lymphoma, and you've got to do the apheresis procedure, and you've got to take that the material, the tissue and cellular material, the lymphocytes, to a manufacturing facility, and then you've got to bring the whole thing back after manufacturing to the patient and deliver it. And it has to be done in a way that you don't compromise the potency of it, the safety of it, and all of that. The fifth point is the longer manufacturing time, because you've got to do so much of work, not only within your manufacturing facility, you've got to do work outside of the manufacturing facility. Therefore, the lead time and the manufacturing time is much, much longer for autologous therapies. And then all of this, the manufacturability issue, the burden of testing, the high level of expertise, which means you have to pay, get very highly paid, compensated scientists in this process, the complexity of the logistics from vein to vein, the longer manufacturing time, all of these leads to high cost of goods. We are talking, in the case of CAR T-cell therapies, we're talking in the $400,000, $500,000 range per treatment. So that is because of all of these factors. So hopefully I gave you a bit of an insight into what are the challenges of using autologous cell therapies. Now let's move to the third challenge that I wanted to highlight, the applications of pluripotent stem cells. And this connects with the roadmap 6 that Jane presented, which relates to disease indications and health economics, and how do you stratify the market, and how do you get into orphan disease status and all of that. So this is a combination of health economics, of course, precise clinical trial design, clinical development, regulatory affairs, and obviously commercialization and business aspects of it. So all of those sort of combine, in figuring out you've got a pluripotent stem cell technology and which disease indication you need to look at, right? So as James sort of talked about the roadmap 6. So what are pluripotent stem cells? Pluripotent stem cells are stem cells that have the capacity to renew and differentiate into the primary germ layers of the early embryo, which would be the ectoderm, mesoderm, and the endoderm, which then gives rise to pretty much all of the cells of the human body. So you've got two kinds of pluripotent stem cells. Traditionally, human embryonic stem cells were used, and we ran into a lot of policy challenges and regulatory challenges early on. And then came Shinya Yamanaka's discovery of taking adult cells from any part of the body and inducing them to become pluripotent stem cells. So in terms of the quality and the properties, there's really biologically, there's not any big difference between embryonic stem cells and pluripotent stem cells. There are companies that are commercializing embryonic stem cell derived therapies also. There are many, many companies that are also working in the area of commercializing and innovating in the induced pluripotent stem cells. So what are the different disease areas that I see where these cells are being used? Diabetes is a very good example where beta cells that produce insulin are being differentiated from pluripotent stem cells. This process is not a simple process. It takes about 3 to 4 weeks to differentiate a pluripotent stem cell into a beta cell that produces insulin. And then, of course, we talked, I talked about CAR T cell therapy and cancer therapy. There are companies that are using pluripotent stem cells to produce CAR T cells, NK cells, and such for cancer immunotherapy applications. In the area of ophthalmology, there are companies that are taking pluripotent stem cells and differentiating them into retinal pigment epithelial cells. And in the case of transfusion medicine, the holy grail is, can we have pluripotent stem cells make RBCs, platelets, so that we can actually stock them up and we don't have to rely on donors. And in the case of rare blood groups, we can even engineer those RBCs to mimic those rare blood groups. So those are some innovative things that are happening in the field. Hi, Jane. Yes.
Yes, one of the things I'd like to emphasize at this point is that there are many, many different uses for induced pluripotent stem cells. Not many of them have made it through clinical trials as of yet. So many of the companies that you're talking about have products in clinical trials, and it's really the new wave of medicine that will go past CAR T, it will go past MSCs and regular cell therapy, because the potential is there. The danger that we're running into with induced pluripotent stem cells is safety. Will they remember to be the cells that you've made them to be?
Correct.
And so that is the step change that we need to address as we move forward with induced pluripotent stem cells. So it's a fantastic new technology that has gigantic implications in the field. But one of the things everybody is really looking at is making sure these cells remember to be what they're supposed to be.
Yes, and there are companies like Pancella that my good friend Dr. Mahendra Rao, a very, very world-renowned stem cell expert, is leading, that have technologies that can actually address the safety issues. And so there are some innovative technologies that are, companies like Pancella are coming out with and partnering with, drug development entities. So the last two examples, and then I'd like to have some questions from this panel. So Parkinson's, very important disease. dopamine neurons are being generated from pluripotent stem cells. And of course, the last is ischemic heart disease, which is a very large market. Pluripotent stem cells are being used to produce cardiomyocytes. So really, there is no end, as Jane said. There is no one, few applications, and we stop there. The potential for pluripotent stem cells to impact human health, in terms of whatever the health condition may be, their pluripotent stem cells have the potential to be able to effect a cure or at least a treatment to manage that condition. So I'll stop there with the top 3 challenges that I wanted to highlight, but also share the excitement of the scientific progress and the innovation that's happening today, and stop there and ask for the panel to ask a few questions or perhaps reflect on some of the thoughts I've echoed here.
Mike.
Yeah, Preveen, interesting things to consider. And when you were discussing autologous versus allogeneic, again, being a manufacturing person, there are some challenges, they are overcomeable, but with autologous, if you need to treat many patients, you have to be very concerned about, number one, the space you have for multiple bioreactors, but more importantly, probably being able to segregate those from patient to patient to patient so that there's no chance for mix-ups. Whereas if we're working with allogeneic products, all we need to do is just get a bigger bioreactor. So, thinking about that, allogeneic is much easier from that perspective to scale up, because with autologous, we literally have to scale out.
Correct.
And there are challenges associated with that. Yes.
Absolutely.
Good points.
Absolutely. But the pain is worth it when the clinical outcomes show that, it works, right? So that's the trade-off, unfortunately, for us. Yeah.
So there's been very few successful allogeneic treatments to date. One is using hematopoietic stem cells from cord blood, has been an allogeneic treatment for patients for many years. That is an example of one excess in allogeneic treatment. It's harder to get over the immunological barriers when you're using allogeneic processes. So finding the right cell types or the right ways to genetically modify the cells so that they do not elicit an immune response is very important. And then if you're doing that, then having a kill switch on those cells so that they don't go and do something that they're not supposed to do when you want them to. That's sort of the state of the industry right now, especially in allo, even in auto.
There are ways, sorry, just to go, sorry to interrupt you. There are ways to actually knock down the MHC class 1 and class 2 molecules, right? And other, immune activating molecules. There are engineering tech, genetically engineered ways of doing that. So just wanted to say that there are solutions, encouraging solutions coming up. Of course, they have yet to be tested, clinically.
The other thing I wanted to mention is with working with Ips cells. If you don't have to put the Ips cell in a patient and you can have them creating the product of choice in vitro, then if you can create enough product, you can then use the end product to treat the patient and avoid the Ips cell problem or safety issue as a whole. So that's very exciting research that's also being done, especially in the area of Parkinson's disease. So I think that we can move on now quickly to Mike and then to Neil. I see we're running over our time, but we have so many valuable things to say. And Mike, please go ahead.
All right, thanks, Jane. I'm going to focus more on the actual manufacturing of various types of products, the facility, in terms of a lot of customers come and ask about two things relative to facilities. One, if they're interested in building their own facility, how do they go about doing that? Or if they want to work with a CDMO, how do they select the best CDMO? And another point I'd like to discuss if we have time at the end is this whole concept of phase appropriate GMPs.
Absolutely.
If you're at early phase versus late phase versus ready to commercialize, how does the level of compliance differ for each of those areas? But let's start out with facility design, facility construction and validation. So say you're a company that wants to build a facility to manufacture their materials that they want to take into clinical trials. First thing you have to consider is how am I going to use this facility? Is this a single product facility? Is it going to be a multi-product or multi-use facility? This has big ramifications because that impacts the clean room segregation that you're going to need. You may have to have completely segregated air handling units for each clean room in that environment in order to keep the potential for cross-contamination at a minimum. Another question you ask, what is the appropriate clean room classification I need? We tend to use the European classifications of grade A, which is the highest, to grade D, which is the lowest classification, or if you want to think it from cleanest to dirtiest. What do you need? What is appropriate for what you're doing? If you're working in fully closed systems, you can certainly get away with grade C, maybe even a grade D environment. However, if you're working in open systems, in flasks, you're growing your cells at very small scale, you probably need to be in a grade B environment because of the potential for contamination of your product. You have to think about what's the scale of my process going to be. I'm early phase clinical, so it's probably going to be relatively small scale. However, what do I want to do after phase 1? Is my intent to go into phase 2, into phase 3, and potentially commercialize, so that I need to be thinking about scalability right now. So let's design that process or develop that process for scalability. If your desire is to just sell off your technology after phase 1 safety testing, I think it may have been you or somebody else mentioned, maybe you let whoever purchases that technology worry about the scale up at that point. Yeah. But that's something to think about. On the same token, when you're talking about scale, that impacts the equipment that you select. Bioreactors versus flasks, open systems versus closed systems, the level of automation needed, that was brought up is potential. As you get closer to commercialization, you want closed systems, you want more highly automated processes at that point. And one of the big things to consider too, as I mentioned initially about a multi-product or multi-use facility, I get a lot of inquiries from people that are interested in, okay, I want to do mammalian cell culture for recombinant therapeutic protein production as well. Can I do that in the same facility as my cell therapy products? We're thinking of making recombinant proteins or plasmids in E. coli. Can I do that in the same facility? I mean, these are all things that need to be considered. Very often what you're going to need for these very different types of products is completely segregated operations for those products. Sometimes segregated not only by separate air handling units, but from my perspective, the company I'm currently working at, we have both microbial and mammalian cell. Our microbial production is a completely separate building on our campus. Not that we don't feel we can maintain the segregation in one facility, but a lot is the perception of customers or even auditors when they come in to look at your facility. If there's even a remote chance that you could potentially cross-contaminate microbes into your mammalian cells or vice versa, that's something you want to, if possible, eliminate, even more than reduce it, eliminate it. Another thing to consider when you're building a facility, there are differences in global regulations in terms of what the FDA expects from a facility versus what the European Union expects, and certainly from what the PMDA, the Japanese regulatory agency, is expecting. So if you're considering building a facility and you're only going to do clinical trials in the US, well, maybe you follow the FDA regs and guidelines. However, if you think you may want to do clinical trials elsewhere in the world, you want to make sure that you're aware of the regulations that may be a bit different for how you design and build your facility and ultimately validate it. So very important thing to consider up front.
Oh,
If
Mike,
you're not,
Mike,
yes,
that's also very important for them. A client of a CDMO needs to ask those questions of each CDMO that they consider.
absolutely. And I was just going to move to how do you select the CDMO?
Yeah,
And you're right on the money. That's one key criteria that you need to inquire about. You need to know where you may intend to do your clinical trials. And if it is internationally, you're right. Make sure the CDMO has facilities in place that can meet those regulatory requirements no matter where you're going to go. Good point. So again, some of the things are very similar that you consider if you're building your own facility. Again, scale. Where are you? Early phase, late phase, commercialization. Make sure the facility that you're going to work with, the company that you're going to work with, has the square footage available, as well as the scale of equipment and the appropriate equipment to run your process at that scale. Another thing to consider from a CDMO, do you want more of a one-stop shop type of environment? Do you want a CDMO that can potentially provide you access to tissue sources to get your cells from? Do you want one that can also develop your process literally from scratch? If you have a bench scale R&D process that's nowhere near ready for clinical manufacturing, you want to find a CDMO that has really good process development and process optimization capabilities, as well as scale up. If you are near commercialization, make sure that they are aware of the regulations that are involved with filing things like BLAs, doing very involved things like process validation, all of which are going to be required to file for commercialization of your product. There are a lot of CDMOs out there that are truly clinical manufacturing facilities that don't have the expertise and the capability for that scale up and commercialization. If your intent is to commercialize, make sure you're working with someone that does, because the last thing you want to do potentially is have to switch CMOs in the middle of your product development cycle. Quality and compliance is, you can't talk about that enough. There are a lot of what I might call GMP light small facilities out there. Depending on the level of compliance you're looking for, you need to assess whether the CDMO you're looking at can maintain and has a quality management system in place that can offer the level of compliance you need. Obviously, we're going to talk in a second about phase appropriate GMPs. That level of compliance will increase as your product development phase increases from early phase to late phase and ultimately to commercialization. So if you're strictly working on a phase 1 product, that level of compliance in some respects could be a bit lower than it is for later phase or commercialization. So again, all questions that you need to ask yourself in order to ask the CDMO that you're going to work with. One of the things that I think is very important in working with a CDMO, and everywhere I've worked, I try to promote this philosophy, is a philosophy of collaboration. There are CDMOs out there, and I'm not mentioning any names and I'm not throwing anyone under the bus. Some of the larger CDMOs out there have lost that collaborative team approach to working with customers. And this is even more important in the early phase, because this is still R&D. They want a project team. They want a team to collaborate with them, to brainstorm, to develop the processes, the methods, to work hand in hand as they take their product through the product pipeline. So having things like weekly team meetings, having a program manager that very closely interacts with the customer is important. So when you're vetting CDMOs, I would certainly be asking that question. Are you going to include us as part of your project team? Will you allow us in your facility to work with you, possibly during the process development phase, maybe even during your phase 1 clinical manufacturing, to be the man in the plant that allows you to come in and work side by side with the team? I think those are very important considerations when you're looking at a CDMO.
Absolutely.
Another thing is from a one-stop shop perspective, it was also mentioned, it's not just about manufacturing, it's about the analytical test methods that are used to characterize your product and used for QC release of your product to the clinical or the commercial environment. So do you prefer to work with a separate analytical CRO and a separate commercial or a separate manufacturing facility? Or would you rather work with a combined CRO, Cdmo that can do both for you? Depending on who you are and your goals, there may be advantages either way. But again, something to consider when looking
Absolutely.
at CROs and CMOs. So with that, let's kind of switch gears a little bit and talk about phase appropriate GMPs, because that's very applicable in this clinical manufacturing stage, which is where most of these cell and cell gene products reside right now. As you mentioned, Jane, not many have actually been commercialized yet, but there's a whole bunch of them currently being evaluated. So what do we mean when we talk about phase appropriate GMPs? What is it? It's basically implies the rigor of compliance that varies from manufacture and testing of early phase products to late phase products and ultimately to commercialization. Now, that can apply to a number of areas. It can apply to your facility design. It can apply to the equipment that you purchase and how you validate it. It can apply to your manufacturing process itself. And it also applies to analytical test methods. So let's talk a little bit about each of those and how it applies to those particular areas. So from your facility design, as I mentioned, there are different global regulations that dictate how facilities should be designed to manufacture certainly commercial products. And there are expectations, maybe not regulations, for how those facilities should be designed for clinical products. My philosophy is for facility design and validation, aim for the highest compliance level possible.
If you can afford it.
Yes, if you can get close to commercial regulations for your facility, do it. This means things like having separate entry and exit airlocks minimally. If you can, have separate entry and exit airlocks for your personnel versus your raw materials. Sometimes that becomes cost prohibitive. Sometimes the space you have available isn't enough. So you can set your procedures to mitigate potential issues if you don't have those things in place. But the higher level of compliance you can achieve, the better. Another one is pressure differentials between clean rooms of the same and different classifications. Again, global regulations dictate different pressure differentials. I would go for the highest rigor possible. Again, makes your facility very flexible. If you do end up manufacturing a product that's going to go into European clinical trials, you already have facilities parameters in place that will allow for that manufacture. So let's talk about equipment. Equipment selection is important, but more so the validation of that equipment. We do validation for early phase manufacturing, just like we do for late phase and commercial. The level of validation is something that is open to interpretation, let's say. So most people that buy equipment for early phase manufacturing do what's called an installation qualification and an operational qualification. I think most of us that live in the validation world know there's also what we call a PQ or a performance qualification. Most people for phase appropriate GMPs will do strictly an IQ, OQ for early phase and start to implement PQ in later phase and certainly for commercial production. Now, what do those things mean? And I'm not going to go into those because that could be an hour-long discussion right there. But minimally, for an IQ and an OQ, you get your piece of equipment in, you put it in its place where it's going to reside, you document everything about where that piece of equipment resides, from the room it's in to the circuit that it's plugged into. Then there's the operational qualification. You literally go through your SOP and say, okay, this equipment, when I turn the on power button on, the equipment goes on. When I set the temperature to 37, I get a readout of 37. Those are very simple examples of an OQ. A PQ is a little bit more complex. So let's use a refrigerator, for example. If I have an empty refrigerator, what is the uniformity of temperature in that chamber? Is it the same temperature in the back upper right-hand corner as it is down in the lower front left-hand corner? Some people might consider that part of a PQ. By the same token, how do those temperatures vary with a chamber that's partially full versus a chamber that's completely full? Those are the types of things that are done typically in a later stage. Most regulatory agencies, including European and Japanese, are fine with IQ, OQ early on and PQ later. From a manufacturing process perspective, I think most of us have heard of the term process validation, a very laborious, expensive set of studies that's done to show the consistency of your product and ultimately of your process and ultimately of the product you're making. Regulatory agencies are fully aware of the extent that it takes to validate a process. Hence, process validation is not expected at early phase manufacturing. However, when you get to phase 3, that's where your process validation studies are kicking. Yes, Jane.
So when you say early stage, are you saying phase 1 and 2?
Yes.
Okay.
So most people will start their process validation when they have their phase 3 process in place. And that's very often where you'll find process changes implemented. People will often do phase 1, phase 2 with the same clinical scale process. If they get to the point where they want to proceed to phase 3 and hopefully commercialization, that's when we really worry about, let's get this thing developed and
Right.
scaled to a commercial scale
Right.
process. Yes.
Right.
By the same token, and this is the last point I'll make, the same holds true for analytical methods. Everybody's heard the term analytical method validation. Analytical methods are expected by regulatory agencies to be fully validated by phase 3 and commercial manufacturing. However, for early phase, we use a term called method qualification that is still a very rigorous process. You develop a method such that it's certainly fit for use. You characterize that method such that you know what is the accuracy of your method, what is the precision, what is the linearity, what is your limit of detection, your limit of quantitation, all those things that you would do for validation. However, at the early clinical stage for method qualification, it is not expected that you have to meet specific acceptance criteria for those analytical methods. Your percent CV is what it is. If it happens to be 40%, but that's good enough for making early phase clinical material, and that assay proves to be fit for use, that's fine. That's not the case when you get to phase 3 and ultimately commercialization. Your product will be well characterized to the point where you understand what you're making, and your analytical methods will be developed and characterized to the point where you know exactly what your precision should be, what your accuracy should be. So again, a lot of interpretation when it comes to phase appropriate CGMPs. And as many people as you talk to, you'll get that many differences in interpretation. So something to consider when either establishing your own manufacturing capabilities or working with a CDMO. Make sure you understand their phase appropriate approach and make sure it's consistent with what you think you need to do for your product.
So many of the things that we've just spoken about is what we really mean by bridging science to business. Because if you don't take into account the IT, the cell type, the processes for manufacturing specific cell types, and then the team that needs to surround that from an executive perspective, you will not be successful in creating your therapy. Which leads to Neil Kellen, who is our token businessman on this panel, who not only understands a lot of the science because he's been in this area for a long time, but really has a strategic approach that I think really allows CellBridge to really not only understand the science and the operations, but build that into a business model. Neil, would you like to begin?
Now, Keith and Preveen and Mike have gone through an awful lot of really cool detail, which reinforces the point you made, Dr. Andrews, earlier in your presentation, is that this is a convergence of technologies in this industry, probably more than any industry in history. And putting all of those things together in a way that works well is really difficult. Not just because the technology itself is complex, but even throughout the life cycle of the technology or the therapy, that complexity changes, okay? So when CellBridge builds the bridge, we also build structures that help companies maintain those bridges. One of the most important things in maintaining that bridge is making sure that there's two-way traffic on that bridge. It's not just science to business or business to science. It's people talking together. Mike talked about collaboration. That is critical in an industry that is rapidly evolving, growing very quickly. You have to be fast. You have to be able to make decisions quickly, and you got to be able to pivot and be flexible. So when Preveen was mentioning that it's one thing to develop a therapy, you then have to get that therapy to clinical level production, which has a whole range of implications. All of the functions and tasks that we've sort of hinted at so far, they don't exist in a vacuum. They all have to hand off to each other. So, you know, using the analogy Keith used on IT is you have systems that need to talk together through a platform. Well, CellBridge, the bridge is that platform for getting all of these activities to synergize, to use a bit of an overused word. And that's a lot of fun to do, but it's not easy. And I think with the breadth and depth of experience that CellBridge has, I think we're uniquely positioned to be able to deliver on that, okay? Now, just like processes don't stand on their own, the business, the science and the business combination does not stand on its own either. So especially if you're a new company, you will have investors, okay? Well, how do you keep those investors comfortable that you are making good use of their money? One of the things CEOs can do, and CellBridge again can help with this, is keep the investors educated into what's going on in the industry. As I just mentioned, and everybody else has mentioned, it's very rapidly changing. It's growing quickly. The companies have to help their investors stay up on this. If they don't, the investors get uncomfortable. And then what happens if investors get uncomfortable? They get a little less generous with their money and they want to hold it back. That's the first way to alienate an investor, is keep them in the dark. People do not invest in things they don't understand, okay? So that's one of the things that a CFO helps with the CEO. And one of the really fun things for me about being a CFO is, if you're really good at your job, you make a lot of efforts at understanding everybody else's work, because you're the person frequently that brings it to the investors in a whole package. And you have to be able to do that in a way that they can understand, which means you have to understand it really well. In helping keep your investors up to date, one of the slides that Dr. Andrews ran through showed the picture of the owl. And we like that picture because what are owls known for? They're known for being able to turn their heads 360 degrees almost, okay? That type of concept of environmental scanning, especially in an industry that's very rapidly changing, is very important. Now, scanning isn't enough. You can see all sorts of things, but if you can't make meaning of them because you don't have either the breadth or depth of experience, you can't translate it to anything that the investors can go, oh, okay, I get it. And I think I understand what that means for you and my money that I've put in your business. Now, one of the things that's often overlooked in especially small businesses, because you're so intent on just getting the work done. You know you've got limited resources, but you got to get the work done. Keith mentioned early in his talk about you start collecting your data as early as possible. as much as you can, start doing everything as early as possible, whether it's collecting data or doing projections, even if they're not great projections, start getting in the habit of doing that. Especially if you're a small company, you need to focus very heavily on cash. You know, I've often asked, well, what are the top 3 things I should be looking at as a businessman? And what I'll say is, are you pre-revenue or pre-profit? If they say yes, I say there's only 3 things you have to look at: cash, cash, and cash. That's really it. Let your operating heads run their business. You make sure that you have enough cash so that they can do what they need. Okay. And there's little things that you can do. Here's a real small example. In one company I worked for, there was a disconnect between the people that signed the deal and the people that implemented the deal. So the deal, as it was signed, called for, and I'm using these numbers for illustration purposes, 100,000 units per month, wrapping up to 1 million units per month in 12 months. They handed that off to the procurement team. The procurement team went off and did their POs and committed to the purchase of all the materials needed. And then I stepped in as the CFO. The CFO changed out and I jumped in, and that was one of the first things I looked at. And one of the things I realized is we're about 8 months into this contract and we're only at about 50,000 units a month, but yet we've already committed to 4 million units. Okay, we've got a potential cash flow problem because we've told these manufacturers we're going to buy all this stuff from them and we don't have the need for it. So there's a lot of stuff that happens behind the scenes that you got to stay on top of. You got to start early. But, you know, I'll just close off by saying, you know, I think one of the fun things in my role with this company is knowing enough about everybody else's work to build the bridge, maintain the bridge, and make sure the traffic is two-way, because that's how you get to translating exceptional science to exceptional business.
Thank you, Neil. That was an excellent summary. And one of the things we do for our clients, and have been doing for one of our clients for over 2 years, is helping them identify their opportunities in the market, and then looking at their internal operations and helping them build their strategies based around that external and internal information. And we've been very successful in helping them grow their company to the point where they're now have done a pre-IPO and currently doing an IPO. And we're still working with them today. We're still acting as their owl, looking at the markets to help them readjust and build their strategies as their company grows. So thank you, everyone. Neil?
I just want to jump in real quick on this client you're talking about. We not only evaluated market opportunities, we gave them a strategy for sequencing entrance into those markets, as well as a pricing strategy, year 1, year 2, year 3, year 4.
That's true, we did. We did. Well, thank you very much. Thank you, Keith, Preveen, Neil, and Mike. I wanted to thank him as well for participating in the Cell Bridge Strategies panel discussion for the World Stem Cell Summit 2022. With that, I'll close, and thank you all very much for participating.
Thank you. Pleasure.